let me just, I just have two slides, but they build on top of each other. So we're going to really pop up the light. First of all, we talk about the cortex interacting with subcortical behavior generators. And like the cortex sits on top of these old parts of the brain and tries to control these old parts of the brain. So this is, I'm going to build up a picture of that right here. So here, this little rectangle represents something that's a subcortical behavior generator. It actually, this orange output here, it drives motor neurons someplace, and you get some sensory input. Examples of a subcortical behavior generator would be the superior colliculus. This is the ones associated with eye movements. The brainstem is associated with a lot of the limb movements and walking and things like that. I mean, and breathing, things like that. The spinal cord. I don't know if the basal ganglia has direct connections to motor neurons. It doesn't really matter, but I put it in here. The point is, there's a bunch of brain regions that are not cortex that actually control neurons that do things, whether it's breathing, moving your limbs, moving your eyes. So I gave examples of eye movements, walking, breathing, reflex limb movements. These are all subcortical behavior generators. It's important to recognize that sometimes these things are fixed. You can think of like a reflex reaction, but they can also learn. The general rules they can learn. So when we talk about the cortex interacting with these, we can't assume that this is some fixed motor system that doesn't change over time. Generally, they do. They can still be reflexive, but they change as your body size changes, trauma, injury, and so on. Okay, so that's what a subcortical generator. So like the superior colliculus is a great example. It gets input from the eyes and some other parts of the brain too, and then it generates eye movements. Mostly non-model based eye movements, like there'd be, reflexive or to tend to some motion, things like that. Now, the cortex sits on top of that. And so let's just talk about the three properties that really matter here. First of all, the cortex projects to these subcortical behavior generators. There's no neurons in the cortex that we know of that project to motor neurons or muscles. You know what I'm saying? I should say muscles. The cortex doesn't control any muscles. The only thing it produces is it projects to subcortical behavior generators, which kind of makes sense in one way because for a couple of ways, actually. One is you don't want to have two different people trying to control muscle movements. The cortex says, move left, and your spinal cord says move right, and what's going to happen. But it's more important to that as will become clearer, hopefully. So the idea is that the columns, or learning modules, Monty, can only affect behavior by driving these subcortical behavior generators, which are much more specific, body-specific, types of things. We have a general system, this cortical column, driving a specific thing, and because the column doesn't know what that specific thing is, right? A cortical column doesn't know what it's projecting to. It's a generic learning module that it has to learn how to drive the subcortical behavior generator. It has to learn to control it via associative learning connections. So this is not a hardwired thing. It has to figure out, how am I supposed to control this thing? So to do that, the column first has to learn a representation of what the thing is doing. It can't just say, if I'm going to tell the eyes to move, I need to know what, well, I'm going to tell, if I'm a column, I want something to happen. I have to know something about the thing that I'm trying to control. I have to know what it does. I don't know if this is clear, but for a generic thing like a learning module or cortical column to control a specific thing about something below, it has to first learn what that specific thing does so that it can control it. Is there any questions about that before I go on? Don't be shy. Okay. Are you still there? I can't see anything.
Yeah, no, sounds good.
Okay, thank you. All right. So let's now talk about vision specifically. So what we think is going on in vision is that the superior colliculus moves the eyes. The eyes, this little arrow is like the feedback from the world. As the eyes move, the input from the sensor changes, and that input comes back up into not only into the superior colliculus, but into the cortical column. And we've proposed that in this case, there are two inputs from the eyes. There's the magnocellular pathway shown in orange and the parvocellular pathway shown in P, with a P in green. And the magnocellular pathway really represents flow patterns on the retina. So the column can observe flow patterns on the retina, which are a direct indication of how the eye is moving. And it can learn, says, okay, remember, the column doesn't know what these cells mean. It has no idea what these inputs mean. It's a bunch of neurons coming in. It says, okay, there's some changing patterns here. I'm going to interpret that as some kind of movement. And I'm going to interpret that kind of movement. And then I will, and if I learn what those movements are, then I will know what the subcortical motor behavior generator is doing. And I can now control it. If this thing can move the eyes left, right, up, and down, and I can see that the eyes move left, right, up, and down, then I can say, oh, I can tell it to do those things. So it's this feedback loop that allows the column to learn how to control this thing. And I want to go into details how it does that. So how I think it does that. These principles probably have to apply to Monty as well. In an embodiment of Monty, any kind of embodiment of Monty, if we're going to have a generic learning module, it's very likely that in some embodiment that there's going to be some subcortical behavior generators. This is our model 3 policies that you've all been working on, plus others, that the learning module will have to learn to control those, and therefore it has to have something like this kind of feedback loop and this basic process going on. Now, I'm going to talk about how this might be done in the neuroscience, which I will say once again, doesn't mean we have to do this way. I'll also say that I don't really have I'm not 100% certain how the neuroscience does this. I have some pretty important clues, and I'll share those clues. But it's still a bit of a mystery. So let's just go into that. So here now is, let me just, how do I hide this? Well, I have this thing on my screen I want to hide. There we go. Here we show a column, let's call it V1. And these little dots there represent neurons, and some of these neurons are in mini-columns represented by these vertical arrangements. So here's an empirical observation, goes back many years. The cells in these mini-columns in layer 5 and 6, we're just going to talk about layer 5 and 6 right now. They seem to respond to movements of edges in particular directions. And so that's just these little blue pictures up here. These are sort of representing the receptive field properties of these cells. Like this would be an edge moving in this direction. This would be an edge moving vertically. This is an edge, the same horizontal edge moving down. And so as you go across the column, you'll see all the different orientations and all the different directions they can be moving. All the cells, I shouldn't say all the cells, but the cells within the mini column seem to respond to that same basic orientation and direction. And the next mini column over represents all the cells respond to a different orientation, slightly different orientation and direction, and so on. In the lower layers of the cortex, these cells all respond best to very long edges, which is what you would want for movement detector, right? You're trying to detect flow across the entire retina. That would be the most important thing to detect for movement of the eye. And we've talked about earlier a lot about how in the upper layers, the cells have similar response properties, but they're much smaller receptive fields, which would be more aligned with the object moving in the world versus here it's the eye moving relative to the world. Does anyone have a question about that so far? Does this make sense?
Sounds good.
Okay.
Yeah.
All right. So I've always wondered, like, why would the cortex have all these cells that respond to the same thing? It's like, what's the point of that? It's like, it doesn't make any sense. And I don't have a picture of this, but in the layer 4, we propose that, all the cells respond to the same thing outside of context, but within context, a single cell will become active, and it forms a unique representation of that item. Something similar, I think, might be happening down here. Let's see here. Whoops, why am I not progressing my slides? Oh, here we go. All right. So my interpretation, this first thing was an empirical observation. My interpretation now is that these mini columns, these represent a basis set of movement vectors. I've already talked about that. Each one is like how the eye is moving relative to the world. In reality, if this is learned, and I think it would be learned, it doesn't have to be so simple like this. It could be, the different movements could be, into the plane, out of the plane. There could be a bunch of different things that are represented beyond just edges moving in horizontal directions. So, we would expect to see I would expect to see minicolumns that don't fit perfectly into this arrangement. And I think there are evidence for that. These are sort of the classic, easier to understand ones. Anyway, so that's an interpretation that these now represent movement vectors. But then a prediction of this theory would be that so now this gets to the point that Jose didn't talk about, this oscillatory interference model for how grid cells are generated. And it's really complicated. I'll show you a picture just to show you what it's about. But what it requires, grid cells require, and I'll show you a paper showing this. Grid cells require that that you have a velocity controlled oscillator, meaning there's cells that are firing at some frequency, and if the animal is moving in a direction, then that frequency increases. Okay, so there's a background frequency, and then if you start moving, the velocity, these cells would say, oh, this whole column would become active saying, oh, we're moving in this direction. And the faster I move, the faster the oscillation of these cells. That's what a velocity controlled oscillator means.
Is it the oscillation or the faster the phase moves through?
Well, it's the oscillation, which will, let me just fill it out next, okay? So imagine it's going at let's say 10 hertz, and now I start moving in a direction. All these cells are now firing at 11 hertz, all of them. And if I go twice as fast at 12 hertz, something like that. But this is also prediction based on theory. I would predict that these cells are all firing at a different phase in that frequency, in that cycle. So they might all be firing at 11 hertz or 10 hertz, but when they actually spike, the individual spikes coming out of the cells would be shifted at different periods of time over that frequency, over a period of the 11 hertz or the 10 hertz signal. So if it's a 10 hertz signal, I have 100 milliseconds, and maybe each of these 10 cells would peak at different points every 10 milliseconds, something like that. They're all firing at the same frequency, but they're phase shifted.
Yeah, no, that makes sense. Yeah, so one way of kind of conceptualizing is like grid cells for one dimension. Like if you were just moving in this direction, just like a grid cell, imagine the spacing is the entire length of this mini column. So the cell at the top is going to fire again after you've gone through all the other cells, and then gone to the top. If you start moving faster, then each cell is going to fire more frequently.
Very, very close, but not quite. Very close. They're all firing at 11 hertz or 10 hertz, but they were trying.
But only when you're moving.
Right. Only when you're moving, but they're all different phases. So at the moment, if I just looked at these cells, they wouldn't tell you where you are. It's when you compare them to a background frequency. So let's say there's a background frequency at 10 hertz, and the base frequency of this mini column is 10 hertz. In that case, only one cell would be peaking at the same peak as the background frequency, because the background frequency is going 10 hertz, and the cells are firing different points in time throughout that 10 hertz, throughout that 100 milliseconds. But only one cell would be peaking at the same point in time as the background frequency. But as I speed up the frequency of the firing of the mini column, the cell that would down peak with the background frequency changes. And so which cell is in coincident activity peaking at the same time as the background frequency will move up this column, or it'll move along here, just like you said, and it'll repeat again at the bottom. And so if I look at which cell is, if I now say I'm going to check my position by which cell is peak, it has the same spikes at the same time as the background frequency, then this becomes like a one-dimensional grid cell module. And you're right, it's only when it's moving, which is a problem, but it is like a one-dimensional grid cell module.
And I mean, I guess even at a certain level, even without the background, it's still kind of like a one-dimensional grid cell module, but just where it's operating so fast that it's hard to read out.
Well, you couldn't read
Because
it out. There's no way to read it out. Who's to say which
unless
cells?
you had like a coincidence detector that was at any given, like when this one spikes, you know, because of the phase difference, they are going to spike at
Right.
different times.
Well, right, but the only way to know which is the right one is you have to compare to the background frequency. If you don't have the background frequency, it's just a bunch of cells that are just spiking, and they have different phases. And so, what can you tell from that? Not much. But it's only when you have a background frequency you can say, oh, there's a coincidence between the background frequency and this thing that I know which is the active one, which one I'm supposed to. Let me show you a picture. How do I get out of here? I'm going to show you a picture here. I get this stupid thing out of my the Zoom thing here. Okay.
Yeah, we see that.
Okay. This is a paper. This is the paper I mentioned earlier. It's called a hybrid oscillatory interference continuous attractor network model for grid cell firing. It's from Burgess Lab. Neil Burgess is an expert in all this stuff. And this figure it has all the information we need in it. Okay. So it took me a long time to understand this figure. These are grid cells, these black circles down here. Okay, these are grid cells. And this is one grid cell. You see my cursor moving around? Hopefully.
Yep.
These, I'm going to tell you, these are the cells in a minicolumn. Okay, and these are the cells in another minicolumn, and these are the cells in another minicolumn. This paper does not make that argument at all. This paper makes a theoretical argument that you need something like this. But I'm telling you, my interpretation, these are the cells in the minicolumn. These cells in this minicolumn all respond to movement in a particular direction. These cells in this minicolumn all respond to a movement in a particular direction. These cells in this minicolumn all respond to movement in a particular direction. And they all fire at different phases, the speeds, all the different phases. So as you're moving here, you go all within like 100 milliseconds or whatever it is. And they're showing that they don't show it here, but there's a background frequency that is also firing, and the red cell is the one the spike of the cell that is in coincidence with the background frequency, and these orange cells are close enough that they also are coincident activity, but not as strong. They're close, they're spiking near each other, meaning this is spiking just before or just after the background frequency, but this is spiking right on the background frequency. So you see a bump of activity going on here, actually. And so the input to a grid cell, any particular grid cell, is derived from this set, right? So a grid cell is not movement direction sensitivity. The grid cell is just as I'm in some location, and what Niels Burgess is arguing is that this particular grid cell gets inputs from a particular set of red cells here, and another grid cell gets a different set of cells. And so if all these cells were doing something very similar to this, each one would activate just like a classic grid cell does, independent of the direction and have all the properties we expect. This lower cell here is essentially an inhibitory neuron or an interneuron, it's a set of them, which are essentially it's keeping these cells active even when the animal's not moving in some sense, right? And so this represents, in the language that we mentioned earlier, the continuous attractor network, meaning it's continuously keeping one of these cells active, or a group of cells active, in the grid cell space. And yet this is the oscillatory interference model, which explains how grid cells, why they change where they fire, and why when I move, there's one cell fire, then the next cell fire, and the next cell fire. Again, this is a complex picture, but you can just think of these cells as a mini column. And that's all I'm going to show from this particular paper, unless there's a question about it.
What exactly is the background frequency?
There's what is it? I always forget which one is it. It's called the beta frequency or something like that.
That's
It's
Beta.
I think.
Stay.
yeah. there's this sort of, these are empirical observations, that there's these frequencies that are sort of continually running in the hippocampal complex and the same in the cortex. And I think they're generated by the thalamus, but I'm not sure about that. So there are these observed oscillations of various frequencies that exist in the brain that are pretty well known. And in the entorhinal cortex, this is beta frequency. I think it's like 8 hertz or something like that. This, let me just show you the basic idea of the oscillatory interference. This is from another paper.
I don't know if it's helpful just to quickly on that figure, just for clarity's sake, like you could have this system in theory set up so that all the VCOs use a rate code. So basically, as you move along, you know, let's say the neuron phi2, it fires with a high rate, and then you move along, and it's not until you move far enough along that it fires again. But
I suppose
it's only because it's encoded in phase, then you need this background frequency to kind of decode it.
Well, no, you still.
I think because
Maybe,
that makes sense
yeah,
in my
right.
head, and I think that just helps to understand this, that it's maybe a bit less mysterious than it might seem. It's basically
Okay.
like you're kind of creating a grid cell that can path integrate in any direction by having these 6.
Right. This is the point you made earlier. I mean, yeah, in theory, you could, like you said earlier, I could have a mini column, and as I move, which cell is active in the mini column, it indicates your location.
Yeah, there's just one cell active, and it's just like
Right,
a firing
right.
rate kind of.
That is correct. That's not what's going on in the brain.
Right.
But that's right. In theory, that's what conceptually, that's what it's doing. That's just turns out not the way the neurons do it.
Right. But yeah, I'm just mentioning that because I think that might help to understand this figure, because then it's a bit simpler. It's just like, okay, yeah, they're basically just like 1D grid cell
Right.
sensitive for a particular direction. And these different cells around the ring, those are the different phases for this 1D kind of, it's almost like it's like a 1D grid cell module. And these are all the different phases.
But for it to get to this to work, to add one more level of complexity on that. The active cell here, this red cell, whether it's an activity or it's a spiking cell, it's spiking and not spiking, or whether it's in phase or not in phase. This has to be anchored. This has to be anchored from an environmental clue. That is, when you enter a new environment, if I recognize the environment, I have to reset these to the same point as they were before. You have to re-anchor these one-dimensional grid cell modules in the same way that two-dimensional grid cell modules are anchored. I just mentioned that it's a complexity of the system, but it's not like these, which cell is firing? How do you decide which one to start with, right?
Right.
It's like, right? It's so you could pick randomly initially, but then as you learn an environment, or in the case of Monty, you learn a model of something, part of recognizing the model is anchoring the location again. And that means picking which of these cells will be active at any point in time. But you're right. I think you said you tried to make it less complex, and I just made it more complex again.
Yeah, no, that's okay. And then maybe just to then quickly recap some like basic physiology, like neurons will struggle to integrate like temporal codes unless there's something to basically help them kind of disambiguate that, like, okay, this is the relevant bit. Like if all of these neurons are spiking at 11 hertz or 10 hertz, whatever, like Jeff was saying,
yeah,
and a neuron that's receiving that information, like how does it know which of those cells that are all sending a spike every 100 milliseconds, which of those is important? And that's where the phase alignment comes in.
If you look at our paper, Why Do Neurons Have Thousands of Synapses, we described that what neurons do is they require a bunch of incoming spikes to occur at the same time, or very close to the same time. So if they're spread out over 100 milliseconds, the cell just ignores them. But if they all kind of arrive at the same within a few milliseconds of each other, then it says, oh yeah, that's it. So that is what I think what you're referring to there, Niels, was this need to disambiguate. It's not just So if you think about how neurons work, where they need this coincident spikes, then this phase thing makes a lot of sense. If you say these are just, you know, some spiking rate, they're all spiking at some, you know, at different points. And how do we differentiate? Different cells have to, anyway, it's like so complicated. I can't keep it in my head at once. But the actual, in neurons, the actual timing of the spike arriving is important. Again, I don't think Monty has to do any of this stuff, but I think it's going to get back to an interesting property that of things we do have to do. All right, it's a good point. Anything else you want to say, Niels? Anyone else?
No, thanks.
Just
Yeah,
another question, or this might be really, I might be missing something really obvious, but while we're not moving in one of those directions, how is it kind of maintained where in that, on that ring sequence we
I don't
currently are?
know. I don't know.
Oh, okay.
That's a good question. See, I've asked myself that question a lot. The evidence we have is that these cells stop firing. Also, they're one directional. So if you go the opposite direction, they don't go backwards. You know, so that's an
yeah,
issue.
I think the implication was the grid cell is all anything downstream cares about, maybe.
Right, right.
And that's what I guess you were saying, yeah, is that like the interneurons and stuff helps basically say if I'm not moving, this grid cell
This cell,
stays active, all the other ones are silent.
this one has the machinery to stay active.
And then
But when I start moving again, I have to start up where I left off.
right.
Right. And
Yeah.
I may be moving in a different direction, in which case I have a completely different set of rings that are active.
Yeah, so I was wondering if it's like a similar mechanism to the interneurons on the grid cells that could be used there, but I think
don't
it's
know.
actually a nice thing that that there is no like reverse of direction, because that way they don't need to learn what's the kind of opposing movement to them, but the grid cell kind of automatically learns that.
Maybe. I don't know. I mean, this is like evolution going out to extremes to figure out how to solve a problem. And its complex solution. But apparently this is what it did. By the way, if you're following all this, a cortical column has minicolumns that go across all the layers. So our cells are organized in a minicolumn layer 4, and layer 3, and layer 5, and layer 6, and so on. And I've come to this basic idea that maybe I should go back to that for a moment here. You can see this now. That there are these essentially a minicolumn within a particular layer is in some sense always doing the same thing. It represents something something generic of which an individual cell represents a specific instance of that. And a minicolumn on its own is not very unique about all. Just like one of these movement vector cells doesn't tell you where you are in a larger environment. But if I were to look at all the active cells in this area, it would be very unique. And the same thing in layer 4. If I look at layer 4, it's representing say an edge orientation. But even if I pick one of the 10 cells in layer 4's edge orientation, it's only 1 in 10 locations. But if I look at multiple minicolumns in that thing, it would be unique. So I think there's this idea of a minicolumn is essentially this processor that goes from a set of cells that are not unique to picking one which is still not unique enough, but then you combine across multiple minicolumns that's unique. Anyway, I don't know if that makes sense, but okay, I want to go back to this. There's a property of grid cells and place cells, which is almost a guarantee that this oscillatory interference system is working. And this is something that's called phase precession. So don't look at the picture yet, just visualize in your head. Imagine you're looking at a grid cell and an animal is moving. And as it moves to a certain location in an environment, the grid cell fires. And when it passes, it stops firing, right? That's what grid cells do. You go into its location, one of its locations, it starts firing, and then it stops firing. When that fire, it turns out that when that cell fires relative to the background frequency changes as it moves through the receptive field of the grid cell. Its phase of firing precesses, meaning as I'm approaching the area where the grid cell is supposed to be active, it fires post the peak of a background frequency. When I'm in the location of the center of the receptive field, it's firing coincident with the background frequency. And as I move beyond the receptive field, it fires prior to the background frequency. That's called phase precession. And in this diagram, we now look at this diagram, we can see what they're showing here is these little, so when the cell spikes at different phases of the background frequency. So what's basically going on here is this is labeled dendrite and soma, but just think of this red line here is the background frequency. Think of it that way. And this blue line is the modulated velocity controlled background frequency. And so they're slightly off. If the animal's not moving, there'd be the exact same frequency. But since the animal is moving, the blue one is slightly higher frequency than the red one. And if you add them together, you get a wave like this. And what this is basically showing is this point right here. If you can see where my cursor is, this is where they're coincidentally aligned. They're right perfectly aligned. So the output, the sum of these two spikes, these two frequencies would be maximum. And so this is an area where the cell fires. And then when it goes into this trough, the cell doesn't fire because these things are misaligned. And this, the physics of it works out that, and this picture tries to show that the firing as you get closer to this point, these things aren't exactly aligned. So it fires, but they start firing anyway, and so it fires when it's not lined up with the peak anymore. It's lined up with the maximum is. I don't know how to describe it better than that, but it's an explanation. This oscillatory interference, it's an explanation why you see this phase precession. And it's kind of hard to imagine how you could get a phase precession like this if you didn't have this oscillatory interference. So this is like the clue that told people that I believe it when it started that way. People noticed the phase precession. Then they said that must be generated by this oscillatory interference. And that came up with this theory about grid cells and how they operate on oscillatory interference. This particular paper was discussing a different flavor. It was saying that the individual dendrites of a neuron, like a grid cell, would be like the circles of cells we talked about earlier. Each dendrite would have its own sort of frequency. I don't think it's right, but that's what this paper is showing. But this gives you the sense of where did this oscillatory interference idea come from? It came from this observation of phase precession. That's a pretty much detailed, yeah.
Out of your interest, what's your concern with each dendrite having that property? Because it seems like, without it, you need a lot of cells just to have one grid cell. And so to have grid cell modules, then you need a lot of cells.
Right.
So it would be nice if that was the case.
Yeah. Oh, I'm trying to go back to my memory right now. Well, it required that these dendrites have some pretty special properties. It required that individual dendrites were sort of like velocity controlled oscillators in particular directions, and something like that. I'm trying to remember now. My memory is poor on this.
That sounds a little bit like the translation invariance thing you've talked about. Like if you move along a certain direction and then you activate certain synapses so that it's
yeah, maybe. I don't know. I have to go back into it.
Yeah.
I felt when it was described, I felt like, oh man, that seems too hard. I don't think that's right. It was more like they're stretching it here. It's like, this is less of an observation, like, oh, we could do it this way. Whereas this picture, someone said, no, they're separate cells. and then I said, oh, well, yeah, but, and I knew what the cortex looked like. It looks like this. So I said, oh, that's evidence that the ring idea was more correct. But I can't prove it. I said, okay, I don't know this for certain. But there was a number of things on the dendrite hypothesis that made me feel like that's kind of squirmy. I don't really,
okay.
how could I?
Fair enough.
But it could be. It could be. I don't know. Let me just finish up here. So we started here. I made these predictions. So if I'm right, then these phase shifting of these, the different phases of these cells firing would be something that could be measured in a lab. And then the other thing here, which is really nice about this hypothesis, and it's not, I can't compare it to the dendrite hypothesis we just talked about, but this hypothesis in general is, in some sense, imagine I have, how many minicolumns do I have in a cortical column? Well, it depends. At a minimum, it would be 100, probably several hundred, depending. Now, as I'm moving, maybe out of 3 out of 10 or 30% of these columns are active, right? Because it's like a bump of activity, right? It's like, this is the most active, and the next one over, and the next one over, a little bit active. So let's say 3 out of 30%, or maybe 3 out of 8, 3 out of 10, let's say 30%. So if I have, let's say I have 200 mini columns, and at that point, 60 are active, right? Because there's going to be repetitions of these. So I might have 60, as I'm moving in a particular direction, I might have 60 mini columns that are active, either strongly active or active prior or prior to it. And I'm picking 1 out of, let's say, 1 out of 20 cells or 1 out of 15 cells to be in phase. So let's even just do 1 out of 10. It would probably be more than that. So I have a 1 out of 10, but then I can do that by, it's like 10 to the 60th. So the number of unique locations I can encode as I'm moving in a particular direction, it would be 10 to the 60th. Tristan, I'm not understanding your comments from earlier, so I'm sorry if I'm not including that. And so that's a really big number. I could encode a very large location space if I were looking at the individual cells that are firing. Once I put them into a grid cell format, it's not so good anymore. I've lost a lot of this property I want. I want a lot of small modules that each one is picking 1 of n, but once I put them in, if I put them in a grid cell module, it's like I've lost a lot of that information. So one possibility, and I don't really understand it. One possibility is that nature said, okay, this method I'm showing you here is really the way that location is encoded. Grid cells are required as a stable storage mechanism to say, okay, yeah, when movement stops, somehow remember where you were, and we want to reignite these same cells again. And so I need some sort of memory to keep track of where I was in this matrix. But this matrix here that I'm showing you, all these minicolumns, fits the bill beautifully for a very high dimensional unique location space, as long as, but I've got some issues with it. So it's not perfect. But it's better than grid cells, because grid cell modules, because I don't have enough. I would have to have a whole bunch of grid cell modules, classic grid cell modules in this cortical column to get it to work, to get a unique location. And we don't seem to see that, but you do see this. And by the way, these cells would appear gritty in some sense, or at least cells that based on it would appear gritty. They'd be like one-dimensional. These are like one-dimensional grid cells in some sense. And if I consider them in phase with background frequency, then I would find cells that seem to be one-dimensional grid cells. And we see a lot of that. We see a lot of movement-sensitive grittiness cells in the entorhinal cortex. I'm sure we'll see them in your cortex too. It's just not a complete theory. It doesn't all make sense, but it's at least the best I am right now. Let's keep going here. So I want to now bump back up to Monty, okay? Or back up to sort of large scale theory. The theory I have is, if you take vision, the magnocellular cells from the retina are center surround receptive fields. If I take those cells and I feed them into a spatial pooler, what the spatial pooler says, I'm going to represent out of these in sort of N and N most common patterns. It's like the spatial pooler always outputs, let's say 3 out of 10 or 3 out of 50 or something like that. It defines a set of mini columns that each represent a common pattern in its input. And so I would naturally, I believe, if I took the magnocellular cells from the retina and I put them through a spatial pooler and I said, okay, I want to represent these by 200 or 100, mini columns, each one being an output of the spatial pooler, it would learn to represent these kind of things. It would say, oh, a common pattern is, these things become active when the cells move in this direction, and these cells become active when moving in this. That these receptive fields could be learned from the center surround RFs of the magnocellular pathway. Clearly, the cortex isn't getting any movement in primates. The cortex does not get movement input from the retina. It only gets these center surrounds, which are not movement. They're just change detectors. But I believe that the spatial pooler would turn them into these receptive fields. And so these essentially are the cortex's interpretation of how the retina can move. It's basically saying, of all the ways the retina can move, I'm going to represent it with these N mini columns, each one representing some sort of, basic one-dimensional vector. I believe that the layer 5A cells would share that same representation. So the layer 5A cells are now encoding the same basic interpretation. They're saying, okay, these cells would then represent, this cell would, layer 5A, this would represent activity of an edge going this way, or the retina moving this way, and this one, retina moving this way, the retina moving this way, or something like that, motion on the retina. And so I have internally now, I have everything I need. I have a way of learning how the retina is moving, forming a representation of it. I then can use that representation to form unique locations in space. And I have my output, which I can send back to my pattern generator. I already mentioned this here, one cell in each phase is sufficient to encode the unique location. So go back to my previous picture here. This orange line is the layer 5A cells saying, okay, subcortical learning generator, you generate some movement of the retina. I have no idea what it is. I observe that movement through these magnocellular cells. They come in as just a whole bunch of bits. I don't really know what they are, but I'm going to run through a spatial pooler. And now I form a representation of a set of basic movement vectors, one per mini column. And now I can send that back to the subcortical behavior generator. It says, when you're generating the behavior moving the eyes left, this is my interpretation for it. So I'm telling you, let's associate my representation with your internal mechanism, which I have no idea what it is. So that way, in the future, I can tell you to move left, or I can tell you to move right, because I've now learned how to control you through observation. That's the upshot of this whole discussion.
This just answers like how the column knows what to communicate to the motor system, but not how it selects which movement to send to the motor system, right? Like it doesn't have the property that knows to get from the location I'm currently at to a different location I want to get to.
Right. Well, layer 5 doesn't know that. Layer 5 is now what we have to introduce next is the allocentric model in the column that can generate behaviors by stringing together these basic movements.
Yeah. Okay.
That's the whole point of this, right? The point of this is you learn this, how to control the subcortical behavior generator by observation. You say, okay, I'm watching you. I think I understand what you're doing. I now have established that I can tell you to do that too. But now I didn't show, I had a picture over here, but I took it out. Now I have a very sophisticated model up here based on my grid cell mechanism, based on my location system, And my model is, I have a model of the world. You don't have that. I have this structured model of the world. And now if I want to achieve something in my structured model, I can string together a set of basic controls to you to tell you how to implement it. You know, you don't know what's going on. I'm telling you, I'm figuring out what to do. And I'm telling you, move this way, move that way. With vision, it's a little less clear that, you know, exactly what a V1 model would be doing. But if you think about manipulating your fingers or moving your limbs and so on, it makes a lot of sense. I might, if this was a somatosensory system, I might be observing reflex behaviors of grasping or, you know, just basic movements of the fingers and the limbs here. I learned how to control them. And now I can string those together for new purposes based on my model, my learned model of the world.
The column
So-
would still have to do a reference frame transform on the motor output.
Right, right. Remember, I didn't show that here too. I took that picture out. Okay, I should have left it in. If we go back to our paper we just submitted, the manuscript we just submitted about the heterarchy paper, in that we talked about the thalamus does the conversion from a egocentric to an allocentric orientation for these two things. And we said that the layer 5A cells, since they don't go back through the thalamus, they have to be converted back somehow to an egocentric framework. And if you recall in that paper, the layer 5A cells are the logical place for the actual happen. They have this unique physiology, this bursting mode, which is very similar to the bursting in the thalamic relay cells. So as we speculated in that paper, that the thalamus does the orientation conversion on the way up, and there's a layer 5B cells project to tell the thalamus what to do, and layer 5B projects to layer 5A. Layer 6B tells it what to do, and layer 6B projects to layer 5A, saying you have to also have to perform an orientation transform. Should I bring up that picture from the paper?
I think it's
Maybe
okay.
that would be.
I don't know if it's not relevant to the main point that you're
Well,
making.
so here we said layer 6B was the orientation of the sensor to the object. We pass it down the thalamus to convert the input from the retina from a retinocentric orientation to an object-centric orientation. But the same conversion has to occur in layer 5A. So this dashed line represents it's already in egocentric form. And I say it's already in egocentric form because it doesn't go through the thalamus. It goes right to the motor areas. And it also then branches up and goes back through the higher order thalamus, meaning it has to get converted again. So these are evidences that this is already in a egocentric format. And then we just briefly mentioned that the layer 5A cells have this unique bursting property, which is very similar to the relay cells here. So without further any explanation, we just threw it out there that it's likely that the layer 5A cells themselves take this input and do the conversion back to egocentric form on its way out. So I think that's what you mentioned, isn't it, Vivian? Is that what
Yeah.
you're talking about? Right.
Yeah.
Right.
Yeah. Yeah, no, this is really interesting. And yeah, in terms of Monty, it's kind of interesting to think about because obviously we've talked a lot about like goal states and in terms of kind of understanding where that falls into this versus like the outputs of columns. It feels like if there is still a place for goal states, that's more like column to column communication.
Right.
Something like L6 or a version of L6 that goes to the apical dendrites in L5 or I don't know, something like that. But then it's the L5 motor output that you're describing here. It's more like what is the neural pattern that I can trigger in a subcortical structure that gives me this movement that I want, that I'm
Right.
now going to observe.
This in the beginning, when I started thinking about this, it was very mysterious to me. What would the layer 5A cell represent? What does it encode? And it was not obvious to me initially. And then now I feel I have a satisfying explanation. It's encoding a language that it observed the subcortical motor generator in a language that the subcortical motor generator would understand. And and it represents movements that occurred, and therefore it is able to control it. And that's also the basis for how we do all of our modeling. There's still a lot of things I don't understand about this, but I think this general idea is a good one. I don't know, maybe I shouldn't show this last picture, but I'll just show it to you. This is my last thing I had on here. This, if you look from a top-down cortex, imagine this is a cortical column, and these individual squares are mini columns. This is not atypical. These circles represent where the bump of activity would occur in those mini columns. It's not 1D like or 2D like you see it in this upper picture. It's where'd my cursor go? I lost my cursor. That's weird. My cursor's gone. It'll come back in a bit. And so you would actually, and there's a lot of evidence for this. We took, like the tank picker is another example, but there's lots of evidence for this. What you'd really see is multiple bumps of activity moving around together, because the mini columns repeat themselves within a cortical column. It's not, there's multiple versions of the same thing. You know, even though I may only be representing 8 different orientations of movement or 10 different orientations of movements, they repeat. And so this is what you see. Actually, if you could see the activity looking down on a cortical column, you'd see these islands of bubbles of activity, and as the animal moves, those bubbles would shift around. This is, you know, I know this is reminiscent of grid cells, but it's not the same thing at all, actually. At least it is a function of the inhibition and creating between cells. Anyway, I just want to point out that there are multiple copies of the same thing going on in a cortical column. It's not just one copy of, you know, there are multiple mini columns representing the same movement vector, which gives us our robustness to uniqueness. Okay, I'm going to stop here, and my cursor's back, and I'll stop sharing.
Nice. Yeah, thanks. That's
Yeah.
I think some interesting things to think about. And yeah, because obviously agree that we want to move eventually towards learning more this kind of action output, what's the appropriate action and stuff like that. And a lot of that's
We don't
hard-coded
have
out.
to, right?
Well,
We don't.
yeah.
I mean, you could, for example, you could maybe come up with a sensor module that maybe, what if we had all cortical columns? Well, I don't like this idea. I said, what if all cortical columns had the same basic motor output representation? Then you send it down to a learning module, and the learning module would convert it from the generic form in the cortex to a specific form for the learning module. I don't think that's a good idea. Another question I had is, when we talk about concepts, like we always talk about, you know, concepts, what is the movement vector? And is it still a movement of a sensor, or is it a movement of something else? It's tantalizing to think that the mechanism in the cortical column really doesn't know, you know, if it's V1, it doesn't know that those are center surround receptor fields on a retina. It has no idea that. It just says, these are some bits coming in, I'll form a representation of them. And so what would be the equivalent from someplace else? I don't know. I really have no idea. Are all concepts rated, you know, built on vector, you know, spaces that are very much like grid cells and, you know, based on coordinates in the real world, or are they getting some kind of movement input that's from some other part of the brain, and it represents a different space? That mechanism I described did not require any specific knowledge about what the subcortical motor generator does and represents. And I also asked, could it be, and I'll leave you this last slide, could it be that what we labeled as a subcortical motor generator is actually a different region of the cortex? The same mechanism could apply. A different region of the cortex could be sending information. I'm a column, I'm getting inputs. I don't know where it's coming from. Maybe it's the motor output of some other column, and I'm going to try to model it. What does that mean? I don't know, but you know, it's possible that some column is just trying to model the behavior of some other column and build a model on top of that. And it may have nothing to do with physical movement in the world. This is the ideas that we've talked about before. with concepts.
Yeah, I think it's really interesting to try and think about movement as these one-dimensional movement vectors that then get combined to form a space. And I didn't really reach a conclusion yet because I was trying to listen to you. So I was wondering if like the findings that they made with bats, if that could be explained if we think about it as one-dimensional movement vectors being combined, and that's why it loses the kind of grittiness in the third dimension, because maybe the kind of up and down movements are
Well,
not.
I don't see why, if I took several hundred mini columns, I don't see why I couldn't represent movements in 3 dimensions.
Yeah,
You
yeah,
know,
you could
but
represent it, but it
if I
might not look
did,
gritty anymore.
well, why not? I can think of one answer to that question. But why wouldn't it? Why wouldn't it be gritty in the 3rd dimension in Z?
But in the case of bats, maybe when they go up and down, it's like a very different kind of movement information that comes in. That's maybe much more sparse than what they get as they.
Well, maybe, but why? Why would that be? I don't know. I mean,
If they go down, maybe they are not moving their wings, so there's like
I don't know. I don't know.
movement.
Right. If you're following all this, here's a potential solution. What if the mini columns really did learn n-dimensional movements, right? Whatever they observe, like we do know that in the entorhinal cortex they can learn one-dimensional movements and they can learn two-dimensional movements. They can perform a grid along a one-dimensional track. We know that they kind of fall apart when they do 3 dimensions in the rat. Okay. But what if the minicoms themselves were able to represent any number of dimensions, let's say 3 dimensions, and they accurately represented that space? But now the problem here is, I have that, remember I showed the top-down view of a column? That's 2-dimensional. The grid cell array that gets locked in is a physical 2-dimensional array. It's not a physical 3-dimensional array. And so I'd have to take my 3-dimensional representation of space in the mini columns and try to force it into a memory that's 2-dimensional. Does that make sense? It's like
I think so, yeah.
If instead of having blobs moving on a 2-dimensional surface, if I could have blobs moving in a 3-dimensional space of cells, then it would probably work. But the cortex isn't 3-dimensional; it's 2-dimensional, and it would force itself into a 2-dimensional array.
Why does it? Why couldn't it work in? I mean, it's just a bunch of 1D movement vectors. If you have more of those 1D movement vectors, they could be in different dimensions.
Right. The problem is
it's
not the 1-dimensional vectors that are the problem. It's the I'm sorry, let me go share my screen again.
Which, yeah, kind of fits with, I guess, what you're saying about like the grid cells being more sort of like a storage point for your location.
Right.
Rather than the core.
This image down here in the lower left.
Yeah,
Imagine
but
this is
why those couldn't correspond to movements in the 3rd dimension as well.
They could. All right, this is not a good. There was this idea that there are many places in the brain where the inhibition is spread horizontally, right? So the continuous attractor network in grid cells. Okay, just think about the continuous attractor network in grid cells. This is the horizontal span of inhibition. It produces a 2-dimensional pattern of activity that looks like this one. It's a different one, but it looks like this. The physical structure of the cortex, if I want to store if grid cells are a storage mechanism for the current location in my space, grid cells are inherently going to be 2-dimensional if they work on inhibition that's in a 2-dimensional sheet of cells. And that's what it appears to be. The neocortex is all 2-dimensional in all of its architecture. So I wouldn't be able to form blobs of activity in 3 dimensions of the grid of the cortical column. I can only form a 2-dimensional sheet of blobs of activity. And so although the mini columns themselves, these guys could represent 3 dimensions or n dimensions. if I want to map them onto a set of grid cells, and the grid cells require a two-dimensional inhibition matrix, then I'm going to force my three-dimensional space onto a two-dimensional memory.
Oh, you mean because
Um,
neighboring mini columns have similar directions that they encode?
Well, that's part of it, but that's not really the issue. The issue is,
I don't know if this is a helpful analogy, but like with CPU design, like part of the issue is they kind of have to design it as a 2D array, because as soon as you add a third dimension, there's nowhere for heat to go. So like,
and it's also very hard. Yes, right.
so
That's
so
part of the
like
problem.
so like all the like transistors in the center of this 3D grid or like cube would overheat. And so
Well, it's not
I don't
a
know if
heat
that's a
issue.
helpful visualization, but
No,
it's
that's
kinda
not,
like,
it's not, but it is, here's a better way of doing it. In silicon design, it's much easier to lay a line down horizontally on the surface of the chip than it is to put a line of a wire that goes vertically on the chip. It's like the process by which they make chips is laying down layer upon layer upon layer. It's not a three-dimensional manufacturing process. And so there's an inherent advantage to, it's very difficult to make three-dimensional wires in a chip. Okay, it's just this way it is. They can't design chips in 3D. They have to basically design layers and put them on top of layers, and they have to connect the layers. So that's a good analogy. Here, it just turns out that the neural tissue by evolutionary design is essentially two-dimensional. And the way the inhibition works is two-dimensional. This could have worked just fine when animals started, walking on the earth or whatever, right? Two dimensions was good. And so whatever reason, this is how it evolved. And so we have this two-dimensional sheet, and therefore these inhibition things are going to be imagine these are areas of activity that are reinforced by, this sort of Mexican hat inhibition, like those local cells reinforce each other and further away they inhibit each other. So this kind of pattern is very common in neuroscience, but it's inherently two-dimensional just because of the nature of the neural tissue. It's not theoretically restricted, but it is in the neural tissue of the brain, it would be restricted.
So then
So
it is
in the-
about the neighborhood relationships of the columns, right?
Um,
If not, I don't see why you can't just put columns with direct.
oh, right. You're right. I'm sorry. You're right. You're actually right, Vivian. I didn't think of it that way. That wasn't the issue to me. The issue, I was imagining inhibitory neurons and what they look like. But right, the way inhibition works, right, assumes that columns, neighboring columns are related, and but I think I could imagine having that same property in 3 dimensions, where instead of these being circles, they were
but I think it's maybe worth pointing out, because I'm not sure if there's an illusion of disagreement or something, because like the argument is basically that, yeah, these mini columns, VSOs, whatever, can do like n-dimensional path integration, which would fit with the fact that animals don't get lost when moving through 3D space. But just if you happen to look at grid cells, for this reason, they only show this really clear griddiness when you look along a two-dimensional plane.
Right, and
Like
so
that's kind of the argument.
right, and so it would lead to problems. Representing 3-dimensional space, it would have. It's sort of it wouldn't be perfect, and I'm just saying that it looks like that's the way the brain. Maybe that's the way the brain does it.
Wait, so
Yeah.
to clarify, are you saying it would be hard to model 3-dimensional space in a good way, or it would be?
It would be hard. I
Grid
think
cell.
it would be hard for biological tissue to model 3-dimensional space. It wouldn't, all the properties you want wouldn't work very well. So for
Wow.
example, you might lose path integration in the 3rd dimension, or you might have trouble re-anchoring in the 3rd dimension, something like that. I don't know.
But that's with like the mechanism that you just proposed with the one-dimensional movement mini columns, or?
That would be the, first of all, in the grid cell literature, you know, most of it has nothing to do with 3 dimensions. Some of it does for bats. Most of it's just like, hey, it represents the map of a room, right? It's 2-dimensional. No one has proposed how this is done, like in Cortex, and this specific idea that minicolumns represent these 1-dimensional vectors. I showed you that picture of the rings. That's as close as you got, right? People saying, oh, there has to be a bunch of cells that have these properties. I actually think probably the minicolumn concepts exist in the entorhinal cortex, but people haven't looked for it. So a lot of this is speculative, right? A lot of this is, you know, me interpreting and making proposals. But my understanding of it would be, I would see why the spatial pooler mechanism should learn n-dimensional spaces. That seems like it would just do that. But then if we're going to rely on sort of a grid cell-like mechanism to keep track of where you are in n-dimensional space, the grid cell mechanism wouldn't work very well in 3 dimensions. And therefore there'd be some deficits in 3 dimensions, whether that's path integration or re-anchoring in 3 dimensions, things like that. I don't know.
But like Niels just said, it seems like we don't have deficits path integrating in 3D space.
That's not true. That's not clear.
Yeah, we talk a lot about this. One thing I can just quickly share while people are getting back on this, like animals in 2D and stuff. I remember when looking into this a while back, there was at least some evidence that animals, even animals that move in 3D, tend to kind of have a planar bias in how they move. So as in, like, if you look at dolphins, this is showing depth of dolphins where they dive. You see how they like kind of go to 400 to 500 meters and then basically spend hours at that depth, and then or minutes, and then they go up to get air, and then they go back down, and then they spend a while. They're not kind of spending loads of time in this kind of like up and down type thing. Of course, that could have other explanations. Like you can imagine that certain animals, like other fish and stuff, also tend to survive at certain depths because of, you know, whatever light and food is there and stuff. And yeah, I just remember when I was at a conference and there was a presentation about grid cells in bats. I think he said the same thing, that they basically tended to fly around this room and stuff at like similar heights. They didn't spend, unless they were going somewhere in particular, they didn't spend loads of time kind of going up and down, up and down, up and down.
You can a little bit of dangerous introspection here. You know, when you're in a room, your head is at some 3D space, the location, right? You have a sense of where you're in the room, but it's not a 3-dimensional sense. It's not like, oh, I'm thinking my head's now 6 feet off the floor, now it's 4 feet off the floor. I mean, you're obviously aware of that, but it's not like, if I say where you are in the room, your first thought isn't, oh, I'm above the table now. It's just, you can somehow deduce that. And that's what grid cells seem to represent in rats. They also did rats climbing on these structures, lattice-like structures, like, you know, little Tinker Toy type of thing. And they tried to figure out, is the rat representing 3-dimensional space? And I can't remember, but I don't remember the details, but the answer was, didn't look like it. Looked like they're somehow hodgepodging together some 2-dimensional representations to make it work for them. It's like here, I mean, in my house, when I go upstairs to my bedroom, I don't have a sense of my 3-dimensional location in the house. I just have a location of where I am in the room. And yeah, I know I'm upstairs, but I don't have this feeling of like, oh, I'm floating above the living room. You know, I don't know. It's just a little weird. And that's all purely anecdotal introspection, not worth much. And I think
wondered. Oh, sorry, go ahead, Scott.
Well, you know, if you can represent a plane with grid cells, that means you can kind of represent a plane that way and that way. And it doesn't really help you with path integration, but you know, if you do have many planes like this, like if you're in a house or something and you have some other sort of more vertically oriented reference frame plane that helps you move through the yeah, through the levels. It still doesn't mean you can really path integrate in like a three-dimensional way, but at least when you want to make vertical movements, you've got some planar type representation that you can use. Yeah, but it seems very hard to recover path integration fully in three-dimensional space.
But I feel
Well-
like it's maybe a bit also about the scale. Like your house is kind of a big space to model. That's kind of like even in 2D moving through a city is difficult to path integrate. You kind of lose it at some point, even in your house. If you think 2D, like if I draw a line from the kitchen straight in this vertical direction, where will I be in 10, where would I be in 10 meters? It would be hard for me to say. But if I have this water bottle, I feel like I have a very three-dimensional model of it, and I can path integrate very accurately on this bottle.
Yeah, but then we show like things like the cube, where if you have a cube and you have images on the face of the cube, you don't really represent a good three-dimensional model of the cube.
But
And
also
it's
the
hard to,
features on the cube, the shape of it, I feel like I also have a very
right,
model of it.
right. Well, I don't know. It's like maybe if I think like, yeah, I don't know, maybe.
Maybe just on.
Well,
Oh, yeah. Oh, what were you saying?
I don't know. I'm just thinking, you know, let's think about Monty. What do we want Monty to do?
Yeah, well, right, because I mean, we have 3D working fine in Monty, but I guess you
Right.
kind of alluded to this like n-dimensional kind of space, and I guess I just wanted to go back to the VSO thing because I've always had this kind of niggling uncertainty about kind of one-dimensional path integration and stuff, and I feel like even with that model that you were showing, Jeff, it seems maybe I'm missing something, but it seems to me like that problem still exists. I don't know if it's possible to pull up the paper.
Which problem?
The so the one with the 6 ring VSOs or VCOs, sorry.
VCOs by Neil Fergus.
What was the problem with it?
So I can explain that by looking at this, but
All right, let me
but
go find that paper.
basically,
Where was that? 1 second.
yeah, no worries.
No, it was in this one here. Try this.
Fergus.
I think it was the
Yeah, the second
55.
tab.
This one here, that one?
Yeah,
Yeah,
that one,
yeah,
perfect.
yeah.
Thank you. Yeah, so let's say you move in this direction, and then you move in this
can't-
direction.
I can't see, you wanna?
Oh, I
how
was
can
saying, so let's say you move right for a while and then
Yeah.
left. And so with these 1D, and for now, let's just kind of, conceptualize them as 1D kind of grid cells or path integrators, because that's basically what they are. So when you're moving to the right, the phi1 would be active. When you're moving to the left, phi4 would be active. If you, and presumably this grid cell that's receiving all these inputs knows where it's meant to be active, like it knows to reactivate because based on which inputs it's getting. Like when it gets either, phi2.
So, well, let's say it's magic. Somehow, when you start moving again, it knows which are the correct
Right.
cells to be in phase.
But so it makes sense in my head. Let's say, so right now phi2 is active, or God, it's really unhelpful that they called it, that's like capital phi and lowercase phi, right?
Oh, yeah, okay.
So
yeah.
okay, I will say, I'll call it module 1 and
Okay.
and cell 2. So module
Okay,
1
great.
and cell 2
Yeah.
is red. So,
Yeah.
right, okay, yeah, and then in module 4, cell 2 is also red. If we move the full kind of phase to the right, cell 2 in
Wait. So, yeah, right. So far.
yeah, if we move a full kind of phase to the right, cell 2 in module 1 will be active again. And then if we move a full phase to the left, cell 2 in module 4 will be active again. So that's fine. And then
Well,
in
it does
that
say-
case, what's happened is like we've moved over, the grid cell's active, we move back and the grid cell's active again.
Well, wait, let's like, what it tells you there is you have a completely different neural mechanism representation if you're going left versus going right.
Right.
Right? You say if you're going left, you have a bunch of with a bunch of these rings are active, and you're going right, a different set of rings are active.
Right.
And so the active neural representation is completely different between those two.
But they, once you've completed that movement, in the cases I described, the set of active cells is the same. And so that's what tells this grid cell to become active again. Like that's the path integration, that you moved away, but
Right,
you moved
right.
back, and so you know you're at the same location. That grid cell should become active
So
again.
you're saying we didn't get our high dimensional uniqueness?
No, this is all fine. Like we're getting, it's a question about path integration. So
Okay,
we're getting path integration when we do a full phase movement away and then a full phase movement back. The
right.
issue I see is, what if we move half a phase, like half the way around the ring?
Yeah,
So now cell 6 in module 1 is active,
yeah.
and cell 6 in module, no, sorry, still cell 2 is active in module 4 because we haven't moved left at all.
Well,
And then-
by the way, my understanding is if you're not moving in that direction, these cells stop being active.
Yeah,
So if you're moving right, then cell module 4 is not active at all.
yeah, okay.
It's a different set of problems.
Yeah, that
Right.
causes problems then if they don't stay active.
Right, you have to somehow reactivate them correctly. Yeah, I mean,
Yeah, and it's I guess my point is, if you just reactivate it based on when it was last active, that doesn't seem sufficient.
no, it's not, right, you'd have to somehow
Because
know how.
otherwise, when you move back left, it's going to
Right.
be the wrong cell that's active, and that grid cell won't activate.
As far as I know, and I haven't looked at this in maybe 4 years or so, as far as I know, no one has addressed that question. I don't know of any, I've never heard a complete theory of how grid cells come about that makes sense. Okay. So, and mine doesn't, the proposal I made doesn't completely solve every problem either. You brought up a good one. That's, it's really complicated. How do these cells know which one to become active? And how does the grid cell, there's a different representation going left and versus right. And by the way, that is actually useful, knowing if you're going, one way versus the other. And sometimes grid cells reflect that. Anyway,
Yeah,
the point is.
Okay,
so you
that's
brought-
fine. I just wanted to check I wasn't misunderstanding something.
No, it's not a complete theory. It just is so damn compelling to look at those cells in layer 5 and 6 that are large field, edge movement detectors, and having all those cells in the Minicolumn represent the same basic receptive field, to think like, oh, that's exactly what's needed, at least in this diagram, the one we're looking at here. That's what's needed. I don't think this paper resolved all those issues. Maybe it did. I'd have to go look at it again.
I always
But I
thought
don't-
the grid cell kind of has to learn how to path integrate between the different movement vectors. So basically it wouldn't just activate with one specific combination of those phases in different movement columns, but it would activate with a bunch of different combinations, but very specific ones. Like,
Right. That's what this requires. And so, one way to think about this is, grid cells anchor based on environmental clues, right? So in this case, the environmental clues would have to anchor the individual cells in the rings. They would have to say, somehow the environmental clues tell me which one of these things should be active. I don't know how that works. We started out today, and I made the observation that this is a very complex area, and I'm going to stop sharing this unless you want to see it still. It's a very complex area. It's not, as far as I know, no one's figured it all out. It's still an area of, as Jose said, of, I don't know how many people are working on it now because it was so damn hard. I don't know if they got enough rewards on it anymore. But as far as I know, it hasn't really been worked out in any sufficient detail to answer all the questions we have. But again, from Monty's point of view, doesn't matter. We can pick up certain ideas from it and try to go with it.
Yeah. Yeah, I feel like for me, one of the bigger takeaways in terms of Monty today was just to think more about the sort of the direct output of each column and how we want to represent that. And because I mean, it's something we've
Yeah.
been discussing recently, you know, even last week with you, but also just kind of generally.
I feel pretty
Yeah.
comfortable with that idea, right? I feel comfortable that it's a learned representation based on what the actual subcortical motor generator did. And therefore it could control it.
Yeah, I
Um,
guess the question is kind of because broadly maybe there's two options. Like one is it's kind of a movement or a displacement, and another one is it's more of like a target, like a location.
mhm.
Like that's how we sometimes conceptualize it. And so like and maybe there's a bit of both, especially depending on what's projecting to what and stuff. But like the location kind of makes sense in terms of like I'm just going to tell you what I want. You figure out how to get it. The movement also has its own appeal because it's kind of like, no, this is actually what you want to do. But that does get a bit closer to almost like controlling a muscle contraction. In
You
some
know, the
way
problem
that
with
it feels like it can put more responsibility on the subcortical structures by just saying, figure out how to get this.
you know, try to follow this logic, see if I can express it clearly. I always thought like, oh, okay, there's these two ways you can communicate. You could communicate like, here's the location you should be at. And originally when we started thinking about the thousand brain theory, that's what I thought. I said, oh, well, you know, we have to communicate the location of this to the location of that. But communicating location is really complicated. And let's not go into the complications yet, but if you communicate a movement vector, it's pretty damn simple, because all I have to say is convert this movement to your orientation and you figure out where you are. Like, I don't know where you are, you know, I'm the eye movement. I don't know where I am relative to the coffee cup. Well, how am I supposed to know that? You know, so, and I can tell you where I am relative to the head, but how do I even communicate that in the language you would understand in terms of location? But I can tell you how I'm moving, and that's easy to translate. And you can figure out, okay, you don't know where you are either, but I'm telling you how to move, and you infer where you are on the object. You have to both infer where you are on the object and your orientation. But that was a problem that was solvable, and we did solve it. And whereas we try to communicate locations, I started there and I felt like, oh, this is impossible. And maybe I just was short-sighted, but it just felt like this is impossible. And then I realized, oh, it's all about movement vectors. That's so simple. It works, you know. Let the recipient figure out what the hell it means. So I don't know how the cortex could tell a subcortical system a location when the models in the cortex don't know locations in the egocentric space. They know nothing about it, and the sensory knows nothing about the locations in the allocentric space.
I mean, I guess that's the conversion we're doing, but yeah.
Is that how we're
Main
doing it in Monty
reason-
today?
We currently communicate locations as input to Monty, but then we use movements for recognizing and learning objects. But we communicate the locations because they're necessary for the voting to work, because otherwise the columns don't know where they are relative to each other in the world.
Right. Well, it feels, if I would ask me, I would guess that voting is a mechanism we haven't really discovered yet. You know, maybe it's related to the colostrum or something. I don't know, you know.
Yeah, and I think we've been saying before that the voting how it is right now is probably too complex, or involves too many reference frame transforms for it
The one we've implemented.
Yeah.
Oh, okay. Damn, too bad. I was gonna say, I don't care. You would do it that way, we'll do it that way. It's fine.
I mean,
Yeah, I
it,
guess it's still
it's not like
question.
a problem right now, but yeah, in terms of at least understanding the brain, it would be nice to find a simpler version, because
I think the
it's
brain-
likely that it's simpler than
Right,
yeah,
I think
yeah.
this idea of communicating movement vectors was a revelation in my mind that simplified it all and said, oh, no, of course, it has to be that way. So
Yeah, I
much
guess if
simpler.
you have an idea of how voting works with that.
All right, well, we can bring that up as a separate topic, right?
Yeah.
I've never really thought about it. Well, we talked about voting, but we haven't talked about how to know your relative positions to each other. That's the key we're
You can
talking
start
about,
with
right?
like with flash inference. You almost have the assumption is almost like there is no movement. Like if someone flashes an object in front of you,
Right, right. Then it's just
um,
it's a bag of features unless you
but
know,
yeah,
right,
but that's
where
the
things
thing.
are.
But humans, like, we don't want to do bag of features because
No, right, no, clearly,
right.
these are not independent. We've known this for a while, that columns need to incorporate their relative positions to each other when it comes to voting. They have to come to an agreement on a common reference frame. I guess maybe the assumption was always that somehow that reference frame is in the egocentric reference frame, and they know where they are there, and then somehow, I don't know, some magic, it happens up in the cortex. I don't know. Something happens that makes it work. So you guys have probably implemented this, so you've thought more about it, but I haven't really thought about the neuroscience much on that one. I could, if you want, I can think about it for next week, maybe. I've never read anything about it. I mean, these ideas, many of these ideas are pretty
Yeah, I remember one of
unknown
the
in
more
neuroscience.
speculative but maybe interesting ideas to tug on more was whether the claustrum could
Right.
be, because I think when we looked at that in terms of connectivity, we saw lots of connections to and from the same layers that tend to be involved in voting, so like L3 and L5.
Yeah.
And so whether there's any evidence that that could be involved in
Well, it seems like we
yeah.
could even approach it from a pure theory point of view. You could just say, okay, here's what we know about cortical columns or learning modules. Let's figure out what they have to do. And,
Right.
you know, what does that require from, you know, mechanisms to do it? Regardless of whether it's a claustrum or something else, you know, just pure, that's the way we usually work, right? You know, we need to have a representational location. How could we do it? Oh, maybe grid cells. You know, it's like, okay, we have to do what exactly? What does the column really have to do to solve this voting problem? It's a bit complex because, you know, it's changing all the time, right? Your fingers are constantly changing their positions relative to each other constantly, which is a much harder situation than, at least it forces you to think about it differently than like the retina. So, you know, I could grab this cup, I can grab this coffee cup, you know, bring my 1,000 branch cup. I can grab my 1,000 branch cup, you know, my hands this way or this way or this way. Oh, I felt that chip again. Surprised me. You know, and they're completely different. The voting is completely different, you know, because their relative positions are completely different every time. Somehow it's got to be calculated very quickly. Yeah, we can think about it. What has to happen?
Yeah, I guess if we did have an answer to that, then we could change Monty to just use movement inputs and outputs instead
All
of.
right, well, how about, you know, I'll think about it. I'll think about it today and tomorrow and see if I come up with something. Please, other people think about it too.
But yeah, I guess it might not solve as pressing of an issue as some of the other topics like modeling object behaviors and stuff, because things work fine with communicating locations right now. So
Okay,
it's
well,
yeah.
that's a good excuse for me to work on it because I'm not doing anything else.
Yeah, maybe
So-
one topic to inspire brainstorming that we were talking about the other day was just that yeah, the kind of idea Vivian had brought up that, you know, maybe behavior is just in behavior columns kind of, or like, you know, there's some columns that just get
Right,
movement,
right.
and so those are just behavior, and then can we get that to work with the things we want, like reusing behaviors for different objects and stuff like that? Does it maybe even work
Yeah.
better than what we had before? Maybe
I
kind of following
wasn't a
that.
fan of that idea, so I haven't spent any time thinking about it. But it doesn't mean it's not right.
Yeah,
At least be nice to know a reason of why they need to be in the same column, because right now there isn't really any good reason I see why, and it would simplify things a lot, and it would make it would seem like such a nice solution, because that way it's really just every column does the same thing, and it just depends on what input it gets, whether it gets.
But does every column do
just
the
as
same thing?
input.
Does every column have the same motor output?
Yeah.
Yeah, I guess
Really?
would morphology models need like a temporal dimension in that case? what would they do with the matrix
Well,
and all that stuff?
there could still be like a state conditioning, like you know how an object can have different morphological states in a sequence. So you could still say, like, you know, when the object stops moving, you learn more morphology again. So that could be the state conditioning for that. But yeah,
I
I
think
mean,
Well,
it's definitely
did-
worth thinking through. Yeah.
It's
just-
worth thinking through it. Again, I think I said once before, like, oh, that could be the whole MT, wear pathway,
yeah,
right?
it's just right now with the current, if we draw our current solution, we're basically drawing the exact same thing 2 times in the same column. So
All right, well, I like it. If I think about it as like the MP wear pathway, then I think it's a good idea. If I think about it like these co-located next to each other, every other column, I don't like it. I don't know why. This is my personal bias.
And I think part of it would be thinking about like what would those long range connections need to be do as well, because I think that was another thing that we also need to solve this even if they're co-located and like behaviors in every column. But like right now the behavior is trying to apply that movement to offset, the lower level column and like how that's happening if that is
Yeah.
happening.
Okay, well, let's.
But it feels like that's worth explaining because it's something new that we haven't discussed, and it's also,
Yeah.
Vivian, like if nothing else, it'll make us more confident that we do need behavior in every column and double
Yeah,
down.
well, all right,
Or-
so I'll retract my objection to the idea. Okay, so.
Yeah,
I'm,
oh, sorry.
I'm just thinking, it's like, this reminds me of so many things. It seems so damn complicated now. But I think in the end, when we figure it all out, it won't be that complicated. It'll be like, okay, it makes sense. But at the moment, it still just seems like, how do I keep all these things in my head at once? MT, colostrum, movements up and down, the representations of behaviors.
yeah, that's what I was kind of thinking when I first thought of putting it in a separate column. It was like, wow, we thought about all of this stuff, all this, like so many things we talked about, but then it might boil down to just having a different sensor module that sends like changes instead of features to the learning module. So-
Yeah. But we do know that even V1 columns have, those receptor field properties I talked about with the directionally sensitive large receptive field cells in layer 6.
And detecting movement in superficial
You
layers.
detect them moving, right? So what are they
But
doing?
that would still be required for the morphology models because you still need the movement input for like path.
Not the movement in the superficial layers.
You mean the movement to L3?
Layer
like,
3,
L-
the movement of the object itself.
But we can double check that because there is classically this idea that MT is more movement sensitive and stuff. So what that's about and stuff.
But
Right.
you would
Yeah.
also still, I think you would still need that input to
Why
compensate.
would I need the object behavior movement in upper layers in a morphology model?
Can I suggest we visit this next week?
Okay, sure.
Just because I feel like everyone's probably starting to flag a bit. But yeah, thanks very much, Jose, for that nice summary of grid cells. And then we can maybe discuss. But I think it would still be interesting next week to talk a bit about the re-anchoring stuff you had found, and at least some of those.
Also, if someone wants to do a quick, like really quick summary of what's known about the colostrum, that might be useful.
Yeah,
I know nothing about it.
I mean, I can at least pull up the same stuff I found before because there was
right. You
a
did
reasonable amount
some
of
stuff.
detail, and let me make a note of that.
All right, we can. Well, I think it's okay. Well,
and then otherwise, I think thinking about. So again, if we maybe dedicate like an hour to grid cell stuff, and then otherwise thinking in terms of behavior, this whole like total separation out of behavior, what that means, if that makes sense with anatomy and stuff.
I think Vivian, you have a clear idea in your head why that makes sense, and I do. I don't know anything you can do to share that. I don't know.
Yeah, I mean it's a pretty small idea, so I'm not sure I can talk a lot more about it, but I could look a bit more into the literature again and see about the kind of
Maybe I'm just trying to understand, like, how would I have a column that just does behavior and a column that just does morphology? I guess it's not
Yeah,
obvious to me.
I mean, if we did want to constrain it by kind of neurophysiology measurements, I think it would be useful to just double check, like, why is it again that everyone kind of says MT is movement, and how much movement is there in V1 and stuff? Because I think MT gets input from V1, so there must be movement in V1 as well and stuff. But
well, obviously the movement has to apply to morphology models, right? So that was part of the thing. By co-locating them in a column, you were able to say, oh, I have this behavioral model, and now I'm looking at a different morphology, and I can apply the behavior to morphology. Easy peasy.
We were doing that using hierarchy, so already using connections between two columns.
We did that hierarchy. I thought we were doing that within a column too, didn't we? Or I forgot.
I think in the main proposal, it was like the higher column would have the behavior model and the lower column would
Oh
have.
yeah, we went in that direction, didn't we?
I mean, we said it might be an option that you could do it within a column, but
I think when we originally had the idea of behavior models, it was all within one column. I think it was
yeah,
back in February.
it's gone. That's
yeah.
what
I
it was like. If
forgot.
I had a nickel for every time we swapped from doing one column for both versus two columns.
If I had a nickel every time I forgot something, man. Dude.
I mean, yeah, if the reason why we need them in the same column is that we need that to apply behavior to morphology, then that I think that would be a good outcome too, to just figure that out.
Right. I think the idea that the where pathway MT is doing this is a intriguing idea. And
Cool.
you know, I'd proposed earlier that it's just modeling the space around the body, but that was kind of a sort of a so-so idea. It wasn't the best idea. So this is something else to think about. All right, you wanted to end this, Niels.
Yeah, I think probably a good stopping point.